Mute. Okay. Oh, okay. Hopefully you guys can hear me online now. So, oh, that explains all the chat bubbles. Okay. So just to catch everyone up, we're interested in this disease, high-risk neuroblastoma, because it's bad. And so we wanted to re-engineer this problem and ask, are there different ways that we can be thinking about this tumor? And in particular, are there targets that we could be going after in this tumor that previously we hadn't thought about? And so both as a postdoc and now in my own independent lab, we've been very interested in doing exactly that. We've done this initially using exome-wide CRISPR-Cas9 dropout screens, where we have used these to try and identify genetic targets that are particularly responsible for driving the genesis and the maintenance of this disease. We've taken these new target maps and actually engineered transcriptional models, which have been very helpful for us to think about different ways to target this disease and in fact several others now. And today I'm going to show you two different stories, one where we've thought about how we could perturb these transcriptional models in using clinically available agents and how that changes cell state. And then in a second sort of story, I'll tell you about some new chemical matter that we've actually engineered that is useful in preclinical models and I hope actually is able to be refined further for treatment of this disease. So just to start with and talk a little bit about this disease and many others. So this is a really simple pie chart that's taken from SEER data. So this is the distribution of cancer in kids under 15 years. And so what you can see is that generally we break things up into leukemias, lymphomas, brain tumors, and solid tumors. And in contrast to the leukemias and the lymphomas, and even some of the brain tumors, definitely not all of them, but some of them, we've made some clinical advancements in those diseases. And now, for example, in leukemias and lymphomas, oftentimes we can cure many of our patients. And now we're in the phase where we're de-escalating therapy. We're taking things away to try and limit toxicity. That is not the situation in the pediatric solid tumors. We are very stagnant. And to a large extent, this is related to this being a very large grab bag of different diseases driven by different genetic events in different tissues. And so just as an example, here's high-risk neuroblastoma again. And what you can see, this is taken from Julie Park's paper in JAMA 2019. So this is overall survival. These are kids who are treated with a single bone marrow transplant versus 2 bone marrow transplants. In fact, this is actually high-dose chemotherapy with autologous stem cell rescue. So it's not a true allo bone marrow transplant. But the toxicity associated with this is enormous. And it bought us about 5% increase in overall survival. So now this is standard of care. This is what we do. Everyone gets 2 bone marrow transplants. So that's bad, right? That's a lot of toxicity. And so just to put an even finer point on it, this is what therapy looks like in high-risk neuroblastoma as an example of a pediatric solid tumor. So our kids get over 18 months, up to 6 cycles of multi-agent chemo. They have surgery, they have radiation, they get 2 bone marrow transplants, and then they get immunotherapy with an antibody that targets an antigen that's expressed on peripheral sympathetic nervous system cells. That, of course, lights you up in total body neuropathic pain. And then they get retinoic acid. So this is brutal. And at the end of this, your survival is a coin flip. Right? It's somewhere around 50, 60%. So you can imagine for the kids who do survive, the toxicities are horrible. And so every survivor of this disease who receives this therapy has hearing loss, kidney damage, heart disease. They develop secondary malignancies, which are usually incurable. They have lung function problems, endocrine disruptions. So therapy needs to be better. Now that I've convinced everybody in the room that therapy is bad and we need to do better, how are we going to get there? Well, here's another big problem is that pediatric cancers, and in fact, the pediatric cancer genome is characterized by a very low mutational rate. So what does that mean? Well, what that means, so here you can see the mutational rate per megabase encoding genes across a whole panel of different pediatric cancers versus adult cancers. And what you can see is that it's really low. And what that means is that we oftentimes don't have a smoking gun oncogene that we can go after. There's no mutated EGFR. There's no amplified, HER2 or ER or any of these sort of common genes. And so we wondered, how do we get at the best targets in pediatric cancer? And so separate from this, a separate group had looked at where are the mutations, the coding mutations that are found in pediatric cancer genomes. And so here you can see in a couple of different types of ALL, in acute myelogenous leukemia, neuroblastoma, Wilms tumor, and osteosarcoma, that you usually see mutations found in a couple of conserved pathways. Things that are involved in transcription and epigenetics, MYC biology, cohesin, and RNA processing. So this concept of transcription being a primary driver of pediatric oncology, and in particular, these solid tumors, was there. And so we got involved in this project called DepMap, which is pretty familiar, I think, to most folks in the audience. For those of you who it's not familiar to, I'll give sort of like a brief outline. And I'll just point out that all the data is publicly available at depmap.org. So, the kind of philosophy behind DepMap is you start with a panel of different cancer cell lines. And in this case, in version 1.0, the idea was that these cancer cell lines reflect some component of the biology of the underlying disease. In version 2.0, which is now sort of in progress, we're increasingly moving to three-dimensional models and to in vivo models to try and validate the findings that we've seen before. So here you take a group of cancer cell lines, you profile their basic genetic and molecular features. So this is whole exome sequencing and RNA-seq, epigenomics. You take that and you send these same cell lines through exome-wide CRISPR-Cas9 screening. You do chemical perturbation screening with thousands of different compounds. And then you take all of this data and you push it together. And you try and use this to actually develop a hypothesis. When cells of this type of cancer have this mutation, they become sensitive to this other gene or to this other compound. And so maybe that's something that we should be studying. And so here's a diagram, again, probably also very familiar to folks in the audience. This is a standard exome-wide CRISPR-Cas9 screen. And in this case, we take all of our cell lines, we make them Cas9 or Cas12a positive. We then infect them with a library of barcoded guide RNAs. We take them through several passages. And at the end, we sequence what's left. And so if you hit a gene that's required for your cells to grow, and that gene is now lost or damaged in some way, then the cells have reduced fitness and you don't see them at the end. So it's a dropout. You're looking for things that aren't there. And so this is what the data kind of looks like. And so here you can see three different genes. Each individual line here is a different cell line. And what you can see is a gene effect score along the x-axis. And the more negative you are, the more likely that gene is to be responsible for a growth phenotype. So here you can see OR4S2. When you knock out this gene, basically nothing happens. So this gene is not required for viability. In contrast, you can see topoisomerase, TOP2A. You knock this out, the whole distribution is left shifted. This is a gene that's required in almost every cell line for growth. And so we call that a pan-lethal. And then there's this third case, ISL1. So this is a transcription factor. And when you knock out this transcription factor, some cell lines die, very few cell lines die, but many of the others actually are unaffected. And this is what we were hoping to find, this so-called selective dependency, a gene that's required in some cell lines and not others. And in fact, every cell line here is a high-risk neuroblastoma cell line, and ISL1 is a transcription factor that drives cell fate in this disease. And so in the first iteration, which we published in Nature Genetics a couple of years ago, we had 84 cell lines that were pediatric in nature, and that was compared against, at the time, about 800 other cancer cell lines. And this is a living, breathing thing. This data set keeps increasing, and again, totally publicly available. And in my lab, we've taken advantage predominantly of neuroblastoma data, but we've also studied osteosarcoma, megaloblastoma, and rhabdomyosarcoma. I'm going to talk about mostly neuroblastoma today, although I'm going to have one slide on rhabdo about the work that we did with Kristen and Haida. So how do we understand transcriptional addiction? So this is another sort of key point. So it's been long enough now, we can probably start talking about neuroblastoma. So this is neuroblastoma is one of that a group of tumors within that very large solid tumor cohort of pediatric cancers. It's a fairly common pediatric extracranial solid tumor, and it makes up 10% of all of our diagnoses and 15% of our deaths. So there's a disproportionate incidence to death ratio. On the I guess right, you can see an MIBG scan. So this is what our patients look like when they come to us. This is not advanced disease. This is when they show up in clinic or when they come into the emergency room. What you're seeing here is an MIBG scan. This is a radionuclide scan that tags tumor cells. Here you can see the very large primary and all of these other dark spots or metastatic deposits. So again, metastasis is the rule. It's not a late finding. This is just what the disease does. This is thought to be derived from neural crest cells that would ultimately go on to form the peripheral sympathetic nervous system that undergo a differentiation. And it has a very low mutational rate. But what we do know is that the most common events are really dysregulation of MYC genes. So we have, in up to a third of cases, amplification of MYC-N, which causes high-level expression of MYC-N. There's alternatively post-translational effects on MYC-N stabilization in non-amplified cell lines. And then about 10% of cases have enhancer hijacking. So there's an enhancer from another gene that's pulled next to the c-MYC gene that drives high-level expression. And so one of the many ways that MYC proteins cause problems at the level of transcription is by binding to upstream low affinity enhancers. And so here you can see in the setting of normal levels of either c-MYC or MYC-N, you have primary binding of this transcription factor to E-boxes and the promoters of accessible genes. And as you drive up expression of MYC-N, it saturates the promoter and it starts to spill over and bind to open regions of chromatin. And this causes massive level transcriptional upregulation, and basically any accessible gene will be upregulated and turned on. And this is exactly what happens in neuroblastoma. And so we started by asking a really simple question. And the question was, what are the genes that neuroblastoma cells rely on for growth that other cancer cell lines don't? And so we looked here at 9 neuroblastoma lines against an outgroup of 350 other cancer cell lines. And now we've done this in larger groups. So we have about 20 or so neuroblastoma lines against over 1,000 other cancer cell lines. And the data looks pretty much the same. And what we saw was an enrichment for genes that were annotated as either transcription factors or nucleic acid binding proteins. And in fact, now as we've looked across pediatric solid tumors, and in fact, even into some brain tumors, we see the same thing. And that is here, some of the most enriched dependencies, genes that are required for viability of these different lineages, tend to be transcription factors. So in Ewing sarcoma, for example, TRIM8, which is a regulator of FLI1 and FLI1 scores because EWS-FLI is the translocation that drives this disease. In rhabdomyosarcoma, some of the top ones, MyoD and PAX3, and in fact, 6-1 score is up there as well, which we'll talk about. So we think this is a common finding, at least by this assay. And so since we had identified these transcription factors as being critically important to cell viability, we wondered, in fact, if any of these were lineage defining or oncogenic. And so as an orthogonal way of looking at this, we thought about enhancer profiling. And so this is a very simplified schematic. Here you can see a gene promoter binding to RNA polymerase, and that bridges to some upstream transcription factor binding to an enhancer marked by some enhancer acetylation. And you can resolve this by ChIP-seq. And here you see, for example, in neuroblastoma cells at the RAD23B locus, you have this little promoter peak, and then upstream you have this little teeny enhancer. And in contrast with that, there are these elements that some people call stretch enhancers or super enhancers, just very, very large enhancers that tend to drive very high level expression of genes. And there is an association with lineage defining genes and oncogenes. And essentially you can see they bind, here you have lots of enhancers, much more enhancer acetylation. And when you resolve that by ChIP-seq here at GATA3, which is a lineage defining oncogene in neuroblastoma, you see it looks totally different. It's just this very large swath of histone acetylation. So we wondered, what would it look like if we looked at a bunch of neuroblastoma cell lines and primary tumors and PDXs? What would the genes be that were always marked by these super enhancer elements? So we did that, and we got a distribution that looked like this. So blink twice because it looks exactly the same, right? And so we noticed that too. we saw the group of genes that were also transcription factors and nucleic acid binding proteins. So what do you do next? You put these two data sets together. And we said, what are the genes that are required for neuroblastoma cells to grow and marked by super enhancers? And we got 10 transcription factors, 9 of which we've now shown are all part of a regulatory circuitry that establishes the transcriptome of these cells. And other folks have done this in many other tumors. So given that we had identified these super enhancer regulated genes that were transcription factors, we wondered if we had actually identified a regulatory circuit. And so what is a core regulatory circuit? This is increasingly being understood in the context of both normal cells and also cancer, but briefly, I'll just say here you can imagine if you have a transcription factor gene that's regulated by a super enhancer and that produces a protein product, this transcription factor can loop back and positively regulate itself and its own expression. And if you had a bunch of these and they regulated both themselves and each other, that's a core regulatory circuit. And it'll give you a diagram that kind of looks like this. And this comes directly from the embryonic stem cell literature, where all of the ES cell reprogramming factors form a core regulatory circuit. And they don't just regulate themselves, but they also go out and they regulate all the effector genes within the cell. And this establishes the transcriptome because all of these transcription factors that are master transcription factors are essentially binding and upregulating gene expression. And this model is helpful because it tells you that if you lose one member of the core regulatory circuit, you should actually elicit a change in cell phenotype. And the reason you do that is that you lose this forward pressure driven by each regulatory factor, each of these transcription factors, resulting in loss of expression, usually of several others. And oftentimes what we see is when you measure this, you see things like death or differentiation when you lose an individual transcription factor. And so this model has been helpful to think about in the context of cancer, where you can imagine in some cases you may have a balanced core regulatory state made up of a couple of different transcription factors. So what happens if you gain a new transcription factor, for example, a PAX3-FLOXO or EWS-FLI or any of the leukemia translocations? These all co-opt transcription and push you to driving transcription through new genomic loci. That elicits a change in the phenotype. Similarly, you can imagine loss of a tumor suppressor transcription factor would lead to change in transcription. And finally, the most common sort of finding that we see is MYC. It's MYC or MYC-N, and they invade these super enhancers and they drive super high level amplification of the circuitry, and this leads to general transcriptional upregulation. And we see this in rhabdomyosarcoma, we see it in neuroblastoma, we see it in megaloblastoma, and many others. And so this led us to wonder, could we capitalize on this and think about targeting this? Would there be a therapeutic window? And so what we did was we went back to our dependency plots and we said, what's happening in the nucleus? Let's look at all of the genes that are interrelated, that are all found within the nucleus, and what's the network that they form? And so we ended up with a diagram that looked something like this. And so here you can see each of these circles represents a gene that's required for neuroblastoma cells to grow. If you're green, you're a transcription factor, and if you're red, you're a druggable enzyme. And we took these and through a lot of ChIP-seq experiments and a lot of knockdowns and degrons, we ended up finding that several of these transcription factors actually do form a regulatory circuit that's invaded by MYC-N. And this is what the sort of adrenergic, so-called core regulatory circuit in neuroblastoma looks like. And so over the last couple of years, we've spent a lot of time and effort targeting this thing, and we've done it through a bunch of different ways, and I'll tell you about 2 of those today. And how do you target transcription factors? Well, increasingly, people are using degron motifs that are found within C2H2 zinc fingers or TRAF-TACs and a number of new technologies. We had initially started by targeting the epigenome. So thinking about targeting histone readers, writers, erasers, and chromatin remodeling proteins. Increasingly, we're using targeted protein degradation to do that. And I'll just put a plug. I love this image of the scientist holding a PROTAC, proteolysis targeted chimera, and taking out the trash, which is, of course, this undruggable target. We use a lot of PROTACs in my lab, but we don't only use PROTACs. So we're pretty agnostic. Anything that's gonna help us out in order to both understand the disease biology as well as kill neuroblastomas, that's totally fine in my books. So I want to now sort of stop and pause and tell you a little bit about a story where we started with a clinical inhibitor that we thought was actually very non-specific. And it turned out to be actually really laser focused and more like a missile. And so we'll go back in history. So in 1985, Carol Teal, Pat Reynolds, and Mark Israel published this paper in Nature where they showed that if you take here neuroblastoma cells in culture and you treat them with retinoic acid, a vitamin A agonist, that the cells completely change morphology and they form these neurites and for all intents and purposes look like differentiated neurons. It turns out that this is actually not just a cell culture trick. This makes a clinical difference too. And so in 1999, Kate Mathey published this paper showing in the New England Journal, showing that if you added retinoic acid to patients with neuroblastoma, you actually achieved a really significant, nearly 20% increase in overall survival. And so patients with transplant and retinoic acid certainly did much better. And if you look, actually, that got us pretty close to 60%, which is where we are now. So this matters. So what does it do? How does it work? So it turns out that because of this MYC-N-driven transcriptional amplification problem, for many years, we actually didn't know how retinoic acid worked. And that was because when you targeted it, you lost MYC-N. And if you lose MYC-N, you lose that transcriptional amplification. And so now you have no idea what's going on at the transcriptome level. And so what we did with Mark Zimmerman in Tom Look's lab was to do a little trick where we added in an external control using synthesized RNAs. And now we had a control that we could compare back to and look at what was happening to the transcriptome overall. And so here what we did was we treated neuroblastoma cells, these two, BE2C and NGP, with retinoic acid for several days, and then looked with this spike-in method by RNA-seq. And what we saw was loss of 3 members of the core regulatory circuit. We saw loss of MYC-N, GATA3, and FOX2B. And when we looked epigenomically, we saw that these loci, so here GATA3 and here FOX2B, they had these large super enhancers, and those super enhancers were lost, and instead they were replaced with histone trimethylation. So we weren't just losing expression, we were completely changing the epigenome. They were losing enhancer material, and now these genes were being repressed. But previously, I had said that when you lose transcription factors in the core regulatory circuit, you usually cause death. And in this case, we were seeing differentiation. And so this taught us something very important, which was that it wasn't just a death phenotype, that you could actually drive differentiation, which played into the work that we did in rhabdomyosarcoma. And so, in fact, we weren't just losing transcription factors, we were gaining new transcription factors. And so when we looked at our RNA-seq data, what we saw was upregulation of 2 genes, MICE1 and SOX4. And when we looked at the loci for these genes here in 2 different lines and showing SOX4 as an example, we had new histone acetylation established upstream of these genes. So we had new enhancers that were driving the expression of these genes. And so what about the genes that didn't change expression? So here, for example, this core regulatory circuit member, HAN2, you can see in the resting case at this very large super enhancer, it's driven by GATA3 and FOX2B binding with really not much going on at MICE1, SOX4, and RAR alpha. And now when you treat with retinoic acid, you lose GATA3 and FOX2B binding because these transcription factors are no longer expressed. And instead, now you gain binding of MICE1, SOX4, and RAR alpha. So what's happening here, we believe, is that you have a regulatory circuit, you're losing some transcription factor members, you're gaining new ones, and that's opening up and giving you new transcription at different sites across the genome to drive differentiation. So we went on to ask, is this reversible? So one of the big problems with retinoic acid therapy in patients is that you give this drug and then we give it for 6 months and then we stop. So what happens when you stop? So what we found was that if we treated our cells in culture for several days and then washed out this drug, everything returned back to normal. So under treatment selection, we had induction of MICE1 and SOX4 and repression of the normal core regulatory circuit. This would just return back to normal when you wash out the drug. We saw this at the protein level. And in fact, we saw this morphologically. You treat for a couple of days and the cells form these neurites, and then you wash it out and they go right back to normal. And in fact, they proliferate the same. It's like you did nothing. So we wondered, is actually the induction of these transcription factors, is that important for this new phenotype, for this pro-differentiation phenotype, thinking that could perhaps give us some clues as to what might be driving this in the first place. So we took these lines and we generated knockouts here. So we knocked out SOX4. It has no effect in the endogenous case because there's nothing driving its upregulation. So these cells proliferate totally normally. But now when you treat them with retinoic acid, you get a blunting of this effect. And in fact, the cells don't differentiate. They shift into apoptosis and they die. So SOX4 in particular, and we showed this with MICE1 as well, is actually required for this self-state change and required for the induction of differentiation. So yesterday I went skiing and it was really fun. And so here's a great picture of me and Kristen and Haida. And the reason I'm showing you this is because we're going to switch gears and we're going to talk about rhabdomyosarcoma for exactly one slide. And I'm going to tell you that what we saw in rhabdomyosarcoma in many ways very closely parallels this story. So as Haida said, they were studying a transcription factor called 6-1. And individually, we had also been trying to study the core regulatory circuit in rhabdomyosarcoma and had found this transcription factor 6-1 as being really important. And so here from just from this paper published last year, what we saw was that many of these rhabdomyosarcoma cell lines, each of which is a blue dot, has very high level expression of 6-1 and is left shifted and dependent on 6-1. And we had around the same time established that many of the rhabdomyosarcoma cell lines used a core regulatory circuit that looked something like this, where MyoD, which is the master regulator of muscle differentiation in normal cells, is actually sort of brought into a regulatory circuit involving MYC-N and several other transcription factors in 6-1. And Jessica in Haida and Kristen's lab had done some really interesting work showing that if you knock down 6-1, that now the cells actually differentiate. Well, this is like a genetic version of what we saw with retinoic acid. And so here you can see in the resting case, based on a DAPI stain, all of these different nuclei. And then when you knock down 6-1, you have this induction of myosin heavy chain, which is a terminal differentiation marker in these rhabdomyosarcoma cells. And Jessica had gone on to show, in fact, that when you knock down 6-1 and then graft these cells into NSG mice, that actually the tumors don't form. And in genetic models and zebrafish, that this is a loss of this gene is actually critically important for repressing tumor formation. So how does it work? Well, in combination with Haida and Kristen, Jessica and I did some ChIP-seq, and then I'm just showing you sort of the punchline, which is that 6-1, we believe, is interacting in some way with MyoD. And so in the setting of normal levels of 6-1, MyoD is present here by cut and run qPCR at a number of stem and oncogenic genes. And when you lose 6-1 expression, now MyoD is lost from those sites and it relocalizes and drives differentiation. And so it led to this model whereby these core regulatory circuit transcription factors aren't just driving their own expression and each other's, but they're actually physically interacting. And as part of that, that is critically important for restraining transcription factors at key loci. And so this is really where my lab is going, is thinking about how can we rewire or reprogram transcription in different disease states. And we've shown we can do it using chemical means and we can do it using genetic means. And in fact, the concept is really straightforward. It's just you have a regulatory circuit that's driving transcription. And if you are able to perturb that in some way, changing the transcription factor members of this circuit, you're going to get a different phenotype. And increasingly, we're identifying that expression is only part of the game and that transcription factor activity and localization, partners, and are all important. And the idea is that eventually by putting this together, hopefully we're able to actually derive clinically useful phenotypes. So in the last part of the talk, what I want to tell you about is a more recent story where we thought we were building something really laser focused and we built a nuclear weapon. So going back to this, so we really wanted to drug our regulatory circuit. And so we knew that every gene within this circuit, within this interaction diagram, was required for viability of neuroblastoma cells. And it was not lost on me that sitting right in the middle and most densely interconnected was this gene called P300. P300 is a histone acetyltransferase. It's got a partner or a paralog called CBP. You can see this is the domain structure. It's got KIX domains that mediate transcription factor binding. It's got a bromodomain that it uses to bind to acetylated protein, including histones. And it's got a histone acetyltransferase domain, which is a misnomer because it doesn't just acetylate histones, it acetylates thousands of proteins within the cell. But the best sort of characterized target is a histone. And you can see these two proteins, their domain structure is very similar. In fact, these are highly homologous proteins and they're co-expressed across many different cell types. And so in the setting of acetyl-CoA, these two proteins have a canonical role where they actually will use acetyl-CoA as a substrate to acetylate histones, and this provides a bromodomain binding site for things like BRD4 that then recruits the transcriptional complex. In fact, they established the H3K27 acetyl mark that we had been using to define super enhancers. So that kind of fit. And so we observed that here across a panel of different neuroblastoma cell lines, that the majority of these cell lines actually relied on P300 for survival, with really only very few relying on CBP. And so that's indicated here. The darker red color means that that cell line in particular died when we knocked out P300 compared with knockout of CBP. So lots more red under the P300 versus the CBP column. And we could validate this in low throughput. So here we knocked out P300 in Kelly neuroblastoma cells, which is a sort of common model that we use. And we saw complete loss of the driving oncogene, which is McN in this disease. And we saw complete loss of histone acetylation at lysine 27, the sort of target mark for this protein. And when we knock out CBP, which is the paralog and is equivalently expressed at the protein level, we saw neither of those things. And in fact, when we did colony assays, we saw the same thing, which is compared to our controls, P300 knockout was pretty toxic to these cells, and CBP knockout had really very little effect. And over the years, through extensive medicinal chemistry campaigns, people have identified a lot of different compounds that target these proteins in different domain-specific ways. Unfortunately, because these two proteins are so highly homologous, everything that targets one also targets the other. And so, but we took advantage of some of these molecules that target either the HAT domain or the bromo domain, and we treated neuroblastoma cells with them, and we identified that by far the most potent was this molecule A485. That's a HAT domain inhibitor, so it's a catalytic inhibitor. And so we wondered, well, why are P300 and CBP playing such distinct roles in this disease? Is it that one of them is mutated? No. We went on and looked through very large sequencing databases, and these two proteins are extremely rarely mutated in neuroblastoma. We knew that they were both expressed, and in fact are coordinately expressed across all of human cancer, and that's based on large, again, database searches. And so we wondered, was it actually where they were binding across the genome? And in fact, it turns out that that's probably what's going on. And so what we did was we performed ChIP-seq in a number of different neuroblastoma cell lines. And here we rank ordered by P300 binding. So up here at the top in these heat maps, you have regions that are most densely bound by P300 down to regions that are lightly bound by P300. And we rank ordered CBP, the target mark, H3K27 acetyl, open chromatin, and then all of the master transcription factors in neuroblastoma. And basically what you can see is that regions that are heavily bound by P300 are heavily bound by everything. Everything kind of looks like it moves together across the genome. But when we looked at the core regulatory circuitry loci in particular, we saw something very different. and that was this. So here you see this very large super enhancer at the FOX2B locus. This is one of the CRC members, and we saw a P300 binding there, but we saw almost no CBP binding. And the CRC loci all looked like this. And so we wondered, well, what was actually bringing P300 to DNA to give that site specificity? It turns out that P300 and CBP both require a chaperone to get to DNA. They have non-specific DNA binding activity. And so we took our chip data and we looked under the top peaks bound by either P300 or CBP. And what we saw was an enrichment for a transcription factor binding site called AP2 beta, or corresponding to TFAP2 beta. And so this would imply that TFAP2 beta is a chaperone bringing P300 to DNA. And if that's true, then it should co-immunoprecipitate. And so we did that. And yes, if you IP P300, it pulls down tons of AP2 beta. And if you IP CBP, you don't see anything. And so that also implies that if you knocked out AP2 beta, that you should lose H3K27 acetyl with no change at all in P300. And so we did that as well, and that also happened. So we think that this core regulatory circuit member is actually escorting P300 to DNA to give it that sequence specificity. But what we really wanted was a molecule that we could deliver that would selectively target P300. And the rationale was if we targeted using a systemic therapy, something that would hit P300, then we would actually lose the ability in neuroblastoma cells for them to sustain transcription because they don't really care about CBP. And in normal cells, you might actually have some CBP hanging around, and that would presumably limit the toxicity and perhaps would give you a therapeutic window. And so to do that, we partnered with Jun Chi, and we used, who's a medicinal chemist at Dana-Farber, and we developed a small molecule called a PROTAC, which essentially bridges your target of interest to an E3 ligase complex. And we used a binding moiety that sort of bound to cereblon. And the rationale is that this brings your protein in close proximity to an E2, which then polyubiquitinates and causes proteasomal degradation of your protein. So it essentially allows your target of interest to get eaten by the proteasome. And so with June, we took advantage of the fact that the most potent molecule was this molecule A485. So we took that and we turned it into a PROTAC, which we called JQ81, which definitely stands for just quells the acetylation domain and doesn't stand for our initials. And so here you can see on one side, the parent compound A485, it's got a linker and then an imide moiety, and this binds to cereblon over here. So really it's this structure here. And the first thing we did was try to model this in order to build the linker to be the right size. And so we did a lot of computational modeling. The problem, of course, is that these proteins are so large that there's no crystal structure for P300 and CBP, and huge parts of these proteins are actually intrinsically disordered. And so it's been very challenging. So what this meant was a lot of trial and error with compound design. But in the end, what we ended up with was this molecule J-quad. And the first thing we did was we biotinylated it in the linker, and we used it in pull down assays. And what we saw was that actually biotinylated J-quad pulled down P300, but it didn't pull down CBP. And we were really struck by this, because the parent molecule binds to both P300 and CBP, but now the degrader only binds to P300. And so we didn't believe this. So we did Silac labeled mass spec. And so we treated neuroblastoma cells for 24 hours with this compound, extracted out the nucleus, and did mass spec. And what we saw was loss of exactly one protein at 24 hours, and that was P300. CBP totally untouched. And we could visualize this using Western blotting as well. So here you can see a time course. Over the span of 2 days, we see degradation of P300. CBP is retained until fairly late. At this time point, though, the cells are really not doing very well, as you can see by our PARP indicating that they're in apoptosis. So we wondered, how does it work? So we went back to this model, and here you can see the PROTAC on one side binds to our target, which is P300. On the other side, it binds to cereblon. And so we decided, well, what would happen if we knocked out cereblon? That should actually remove compound function. So we knocked out cereblon, and we tested looking at 7 days of viability. And what we saw in this dose response was that we could completely rescue the effect of the compound. In fact, we went on to do very large screens with this compound and demonstrated across hundreds of different cancer cell lines that those that were sensitive were cell lines that had very high level expression of cereblon. And so we took cells that were actually insensitive to this compound, we overexpressed cereblon in them, and now converted this into an active degrader. So here you can see a neuroblastoma cell line that in response to JQuad1 has no effect at all on P300. When we overexpress cereblon, in fact, we restore this. And independently of this, we had been doing some work with Paul Gielaher and Jun Yang's group at St. Jude, looking at mediators of specificity and sensitivity, and actually, again, pulled out that the #1 top hit was really cereblon. So expression of this is actually, we believe now, almost even a biomarker of activity. And so what does this do to cells? So we treated neuroblastoma cells with this, and here you can see in these dot plots, which represent H3K27 acetyl spike in ChIP-seq. And so here at 0 hours compared to 6 hours of treatment, you see not much. Each individual dot is a different enhancer peak, and most of them are sort of mean-centered right around 0. And at 6 hours, we really have almost no effect at all on P300 expression, so that's to be expected. At 24 hours, we start to lose P300 expression, and now we've essentially wiped H3K27 acetyl off the genome. And when you look at a micro scale, so here at the hand 2 enhancer, you can see it looks like we ran over this enhancer with a lawnmower. I'm trying to make that a thing. I don't know if that's funny. We paired this with RNA-seq, and we see something very similar, which is enhanced loss of transcription at super enhancer marked genes, of course, at the RNA level. As you can imagine, cells don't like that very much. And so here you can see a MYC amplified neuroblastoma line treated with JQuad, a stereoisomer of JQuad, or the parent molecule A485 in JQuad causes the most suppression of growth. This is, in fact, apoptotic cell death as marked by cells in sub-G1 phase, as well as by caspase and PARP cleavage. So this is really interesting. Here you inhibit the protein. In fact, you inhibit 2 proteins, and the cells actually freeze in G1. Whereas here you degrade the protein and the cells are dying. And so we wondered, was there in fact a scaffolding function in addition to a catalytic function for this protein? And so one of the ways that neuroblastoma cells repress apoptosis is by having extremely high levels of MYC-N. So we wondered, was P300 interacting with MYC-N? And so we did pull downs. And again, yes, P300 pulls down MYC-N and CBP does not. And when you degrade the protein, you see loss of MYC-N expression. Whereas when you catalytically inhibit it, you have no change in MYC-N expression. So we think in neuroblastoma that actually P300 is forming both a scaffold to bring MYC-N to chromatin as well as establishing enhancers, and that in fact both roles matter. So you might say, well, why isn't this just all MYC-N then? You're losing MYC-N, that's the driving oncogene, that's really what's causing apoptosis. So we wondered that too. So we overexpressed MYC-N in these cells that already expressed tons, but we did this using a retroviral construct so that it wouldn't actually be sensitive to P300. And what we saw when we treated cells with JQuad was not a big change in terms of sensitivity. And we went on to perform SLAM-seq, which is a nascent RNA sequencing technique where you label with modified uridines and then do an RNA extraction to sort of see what's happening with nascent RNA. And what we saw was that catalytic inhibition with A485 really had very minor effects, but that JQuad was causing a nearly 50% reduction in overall gene expression across the entirety of the nascent transcriptome. And that included a increased representation of genes that were required for viability of neuroblastoma cells. And so this is the fun part where I get to show you how it's a nuclear weapon. So this is the interaction plot. Again, each of these different genes is required for viability of neuroblastoma cells. And I'm going to put a circle around every gene that's transcriptionally lost at the nascent level when you treat with JQuad, and not when you catalytically inhibit both P300 and CBP. And it looks like this. So every one of those genes is lost. That's why we think it's a nuclear weapon. So we took this first generation molecule and we wondered, Would it actually work in vivo? So we grafted, xenografted into NSG mice, and this first generation molecule, which is really a probe, is not fully optimized, has solubility problems, still caused a significant delay in tumor growth. It caused prolongation in animal survival. But what I was most struck by was the toxicity profile. And so we did extensive toxicity testing with this molecule. I'm just showing you animal weight, which you can see looks the same, but we did blood counts and liver function tests, kidney function tests. We took out all of the organs and looked for any evidence in immunocompetent mice of any fibrosis or lymphocytic infiltrate, and we found no difference. This compound was inert. Except when we looked at the tumors, we could see loss of P300 immunostain with retained CBP. We saw in these tumors, by doing spike in RNA-seq, we saw an enrichment for loss of MYC target genes. And when we looked at the CRC, we saw downregulation of all members of the core regulatory circuit. So essentially, what I hope I got across today is that lineage-specific transcription factors are key targets, that they form these circuits that establish the transcriptome, are dysregulated in cancer, and actually are potentially targets for therapeutic intervention. And so we started with this clinical problem, and we used that to define these new targets using exome-wide screening. And in fact, we're doing this in many other diseases now. We use that to build these transcriptional models. And now by interrogating those transcriptional models, I think we're getting deeper insight into ways to change cell fate. And as you can imagine, my lab now is sort of moving towards better targeting. We're trying to improve JQuad. We're trying to combine it with other therapies. We're increasingly moving into immunocompetent models and, of course, thinking about chemoresistance. And so I don't know if you can see that. It basically says science is a team sport. So, of course, there are lots of folks to thank and lots of sort of funding agencies, and in particular, Jun Chi, who has been our sort of medicinal chemistry arm, Brian Abraham, who's been our computational biology arm, all the folks in my lab who do the work, and of course, Haida and Kristen for all the rhabdomyosarcoma work. And of course, our patients who put their trust in us that we're going to do something real with this, with their money and their lives. So with that, this is supposed to say, come join us. So if you're interested, send me an email. And with that, I'll stop. And here's your survey. So I was like, okay on time. Yeah.
For the 6-point interaction with the thing, it would have reduction in binding.
Yes. Yeah. Yeah. I don't want to take away from Annika, who's in the back, who's studying exactly those questions. So all of these are possibilities. So we know, for example, there's a recent paper on SOX11 that is in press now from Frank Spellman's group. And what they've shown, it's up on bioRxiv. So what they've shown is that SOX11, one of the ways that it works in neuroblastoma is by actually recruiting the BAF complex. So in sort of an analogous way to AP2 beta recruiting P300, they have another core regulatory circuit member that can recruit in the BAF complex and establish those open chromatin sites. So you're absolutely right. It doesn't have to be a physical interaction. Although the more co-IP mass spec that we do, the more we're finding that these things really do form protein-protein interactions with each other. So, yeah.
And the other question was for the drug and the safety studies.
Yeah. Yeah. Yes. Yeah. So what we did, so in part, there are, it's a probe, so it still has a lot of PK issues. So it doesn't cross the blood-brain barrier. So that helps us because I think degrading P300 in the central nervous system is not going to be good. The tissues where it's most concentrated because of first pass metabolism is in the liver. So we took out liver and we looked and we saw no evidence of fibrosis. So it looked good. When we stained in the liver, we saw loss of P300 and we saw retained CBP. We also did mass spec PK or NMR PK in different tissues to look to see is the compound taken up into those tissues. And so we saw pretty high uptake in the liver, we saw uptake in the kidney, and in all of the tissues where it's there, we see degradation of P300 and not CBP. But we saw no effect on heart tissue. And when we looked, the compound it just wasn't getting taken up into the heart. So I think a lot more to sort of be done there. And one of the things that we're doing now is looking at tissues where it's taken up to see where is CBP using CHIP instead of using sort of like Western blots or immunohistochemistry. And what does the enhancer profile look like? Is it true that they can actually really substitute for each other in these different scenarios? Yeah, it's a really good question. Yeah. 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 Yeah, I mean, I think we have a lot of theories, but nothing sort of like proof. I think that as cells undergo that cell state change, I think that SOX4 is actually required for that cell state change. I think in the absence of it, we see localization of these complexes to regions that are SOX4 that don't have like SOX4 binding sites. And so I think we're getting partial differentiation, and the sort of output that we're measuring is apoptosis. But I think the reason is that they're not going to where they need to be. Yeah. 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 Yeah, we haven't exhaustively looked. I suspect that there is some old data that suggests that the genes that are dominantly regulated by P300 and the genes that are dominantly regulated by CBP in a couple of different cell systems are different. So Laura Banazinski has actually recently put out a paper showing that in ES cells, P300 tends to be bound at regions that promote sort of like a stable pluripotent sort of state, whereas CBP tends to be more sort of like differentiation genes. So it could be that they're binding to different sites. It also could be, just as equally, hey, one of them is interacting with mediator that's got higher levels of like Med1 or Med9 or something. And I think personally, I don't know, and I think the field doesn't know either. But using these tools, I think we can start to dissect out those. Yeah. Yeah. Hi, do you? Okay. Yeah. Yeah.
So when you use your nuclear bomb, you're targeting majority of all the transcription network that you showed right there. So theoretically, right, growth is they're essential for outgrowth.
Yeah.
Right. So when you treated that at the endpoint of your tumor, you see your tumor, you still see production.
Yeah.
Working.
Yep.
But they're still growing. So
yeah.
I'm curious, do you think there's already other transcriptional circuit that is driving the growth of these tumors?
Yeah.
Or is it just like residual amounts of low level transcription still occurring enough to just barely get these?
Yeah. Yeah, I think that there are a couple of things going on. I think one is really a PK issue. I think that the compound has real solubility problems. And when you look in the tumor, the majority of it looks P300 completely deficient, but there are pockets that have retained P300, and they tend to be more in the center of the tumor. And when we do a Ki-67, we see that those regions are lighting up. So I think part of it is penetration. I also think it is possible that there are sort of secondary mechanisms that are kicking in. The one thing that we know that it's not CBP. So we've taken cells where we've knocked out CBP and grafted them and then treated them with J-quad in the same way, and they have the exact same growth phenotype. So So yeah, it could be either of those things, but I think PK plays a really significant role here. Yeah. Yeah. Haida.
That question about the differentiation therapy. So you show that the difference over
Yep. Yep.
Normal cells clearly are not. And so why do you think that is? Why do you think cells are depending on the user computation that can turn normal tissue that doesn't differ?
Yeah. Yeah. It's not, so I think in postnatal growth, it seems like cells tend to make a choice. That's not exclusively true across all of cancer. So for example, myeloma cells are totally dependent on P300 and totally dependent on CBP. So they have non-overlapping functions. Yes, so I think in neuroblastoma, we're seeing a deep enrichment for one versus the other. There are other lineages as well that do show that. And then there are lineages like AML, for example, happens to have a very high dependency on CBP and not on P300. So I think that there's a lineage specific choice that's mediating one way or the other, because if you go back and you ask, well, what is the phenotype of the P300 knockout mouse or the CBP knockout mouse? They're both embryonic lethal, and they're embryonic lethal at different times. And so P300 knockouts, those mice die earlier than CBP. It's like 9.5 versus 11.5. And so the only system where people have really deeply dissected what is the function of one versus the other in a tissue specific way is actually in hematopoiesis. And so a postnatal conditional knockout of P300 ablates your T cells and has no effect on B cells. And CBP is the opposite. You lose all your B cells and not your T cells. So I think that there are lineages where that choice is actually is critical, and it may be expression in normal tissues. It's just we don't see that in cancer. So I think lineage plays a huge role here. Yeah.
And my other question was sort of your thoughts on the idea of differentiation there, like you show retinoic acid completely reversible.
Yeah.
Which we talked about a little bit.
Yeah.
Then does that mean you're in trouble with any differentiation therapy because the second you take it away, these cells are going to come back?
Yeah.
And does that also mean we get less side effects though, because we can remove any premature differentiation that we want to occur? I mean, what do you think about the viability of that as a therapeutic intervention?
Yeah. Yeah, I think it's really, again, I think it's actually really cell type specific. So in APL, as somebody brought up, right? I mean, it's very clear that you get differentiation. Our patients will get differentiation syndrome, then everything lyses. So it is a differentiation therapy, but along with that, the cells are dying at the end. In this case, in neuroblastoma, we're a bit stuck, because the compound that we're using, retinoic acid, has toxicity, and we treat for 6 months and then we stop. And so the reversibility would seem to suggest that, hey, why don't you just keep it going? The problem with doing that is the length of the trial that you would need to do and the number of patients in order to test that rigorously. It would be really challenging. So one approach that several folks are thinking about is, are there ways to use either synthetic retinoids, tamibarotene or other things that actually typically have worse toxicity profiles than retinoic acid, but shorter periods to drive irreversible differentiation? And then of course, you always worry about the progenitor and stem cell pool, and what that might do in, any number of different conditions. So, but yeah, so I think that's a very complex question and something that we should sort out in rhabdomyosarcoma. So.
Okay.
Cool. Thanks, guys.